So now what I'm going to do is animate meiosis. So got my starting chromosomes here, same as before, nuclear membrane. Beginning of meiosis, the chromosomes start to separate. Nuclear membrane goes away. But then an interesting thing happens. The homologous pairs find each other and pair up, and they lay literally right on top of each other. They don't just get close, they actually bond end to end. At this point, they're sorted, and I want to point out that this represents a diploid set of chromosomes because there are mom, dad, mom, dad, mom, dad, dad, mom chromosome pairs here, and that is the nature of being diploid. At this point, the chromosomes cross over because they're exchanging DNA. That will make sure that the sister chromatids are not identical anymore, because dad DNA is going to end up with a mom chromosome and mom DNA is going to end up with a dad chromosome in each set. So once they've crossed over, then they line up. And in the first meiotic division, the spindle fibers pull, but they don't break any centromeres. They just separate the homologs from each other. So the homologs go to opposite ends. This one came apart already. Okay, so there we go. And you'll notice I got a blue and a pink here on this side. It's random. You have no idea which side's going to end up with more mom or more dad DNA. So the homologs separate. And then right there at the end of that division, a separation occurs. For plants, it'd be a plate, and for animals, it's a pinching process where they pinch in half. Immediately then, the chromosomes line up again. I'll line them up this way. And this time, the spindles, when they pull, they're going to separate the chromatids, the sister chromatids, from each other. I want to point out now that this is a haploid set of chromosomes here where my hand is, because there's no longer a pink counterpart to this blue or sperm-originated blue chromosome. There's an egg purple chromosome here, but the blue one is over there. So this is a haploid set. There are individual chromosomes. There are no longer mom-dad pairs. Same thing here. So now the spindle pulls and the chromatids, the sister chromatids, separate. And what we end up here is with haploid sets. Okay, so we had a haploid. The cell here was haploid. The cell here was haploid. These sets are haploid. What happens now is a nuclear membrane forms around each set. So we've got a nuclear membrane there. We've got a nuclear membrane here. And we got another nuclear membrane here. And another one here. And then the cells will split in half to form 4 cells. We have 4 cells. They're haploid. And because of the crossing over and the way the chromatids separated, none of the cells are identical to each other. There's a variety in each cell, a variety of different gene combinations. And that's the end of meiosis.